Dear students of dentistry faculty, today I am going to present you our 6th lecture. I am Əyyubova, senior lecturer of Histology department. The topic of our lecture will be the integument, development, structural parts, functions, skin appendages, hairs, nails, sebaceous and sweat glands, innervation, vascularization, age features. Then the second part of the lecture will be about the respiratory system, its development, structural parts, morphofunctional features, respiratory portion, air-blood barrier, innervation, vascularization, and age change. So what is the integumentary system actually? You know, the integument, it is the largest and heaviest organ of the body that weighs approximately 15% of total body weight. It has average surface area of about 2 quadrat meters. It comprises the skin that covers the body, then the appendages of the skin. They are hairs, then the nails, sebaceous glands, and sweat glands. The skin covers the entire surface of the body and becomes continuous with the mucous membranes of the digestive, respiratory, and urogenital systems at the external orifices. Skin is composed of the 2 layers, as we can see here. The epidermis, which is the outer stratified squamous keratinized epithelium, and then the dermis, it is connective tissue component of the skin. Then they are separated from each other via the basement membrane. The junction is not a flat plane as we are looking here, and the dermis forms cone-like and ridge-like elevations. The dermal ridges, here we are looking at them. They also call dermal papilla. Then the epidermal ridges and dermal ridges, they together are known as the rete apparatus. Deep to the dermis is a fascial layer that is a hypodermis, superficial fascia, which may contain considerable amount of adipose tissue, and it's in overweight individuals. But the hypodermis, it should be understood that hypodermis, it is not considered to be a component of the skin. So skin includes just these 2 components. The skin, it provides a lot of functions, and the most prevalent there are, first of all, formation of the supple cover for the body, then protecting against impact and abrasion injury, bacterial assault and dehydration, absorbing ultraviolet rays radiation for vitamin D production, then receiving information from the external milieu. As you know, the skin, it is large surface area for the location of the free nerve endings, sensory nerve endings, which may form here free branchings, and they also may form capsulated nerve endings, as one of them we are looking here, Pacinian's corpuscles, which are receptors for the pressure and vibration. And then the next function is excreting of the sweat, then producing melanin, protecting the deeper layers from excessive UV radiation. The presence of raised ridges with intervening grooves in the forms of loops, holes, and arches that are their dermatoglyphs or fingerprints on the pads of the fingertips and toes provides for a less slippery surface so that the smaller objects may be held more securely and provides sensory input for the identification of the object being handled. Epidermis, it is the outer layer of the skin. It is avascular and receives its nutrients via diffusion from the capillary network of the dermis. And here we are looking at the non-hairy skin, which covers the soles and palms, and here we are looking at the hairy skin, which covers most of the rest of the body. And now let's look at this illustration here. We are looking that the epidermis is composed of the stratified squamous keratinized epithelium, and its thickness is approximately 0.1 millimeter, and also on the palm of the hand it may reach the thickness of the 1 millimeter in thickness. We'll look here. That is the thick skin, and but in the sole of the foot it may be even 1.4 millimeter in thickness. That's why there are two types of the skin. That is a thick skin. It present on the palm of the hand and the sole of the foot, and it's hairless, has no erector pili muscles, has no sebaceous glands, although it does have sweat glands. But the thin skin, here we are looking at the thin skin, it present on the remainder of the body, possess hair follicles, erector pili muscles, sebaceous glands, and the sweat glands. If we are looking at the next slide, here we can see the layers of the epidermis. So the epidermis has 5 layers in the thick skin. They are stratum basale, stratum spinosum, stratum granulosum, lucidum, and the corneum. In thin skin, we don't have the stratum lucidum. That's why thin skin will include just 3 to 4 layers. Let's look at the internal structure of the epidermis in its schematic representation. Here we will see the types of cells which present inside of the epidermis. So the 4 different cell types compose the epidermis. Here we are looking at the layers of the epidermis, and that is the dermis that is underlies the epidermis. So what are these cells types? First of all, keratinocytes. They are pinkish colored cells which form vast majority of cells. Then we are looking at the Langerhans cells, then melanocytes and Merkel cells, of which keratinocytes are the most populous and are the ones that are derived from the ectoderm germ layer. The other 3 cell types are distributed among the keratinocytes. And also because the cells of the epithelial surface are desquamated, here you are looking, the lost cells are replaced by mitotic activity of the keratinocytes or the stem cells occupying the deeper layers of the epidermis, and it is believed that epidermal growth factor and interleukin-1 alpha induce mitotic activity of keratinocytes, and transforming growth factor is it's believed that it inhibits such activity. Cell division occurs only at night, as you are looking here, the mitotic division of the cell, and the newly formed cells push the cells above them toward the surface, eventually to be sloughed off from the surface. It takes approximately 1 month for a newly formed cell to reach the free surface and to be desquamated. As keratinocytes move towards the free surface, and they undergo cytomorphosis, which permits the epidermis to be subdivided into these 5 layers just we mentioned. Only 3 of these layers are evident in thin, 3 to 4 of them are evident in thin skin. And now we will characterize these cells. First of all, Langerhans cells. As we are looking here that they are the largest cells of the epidermis. They are antigen presenting cells, and they provide defense of the epidermis from the foreign invaders, and that are these cells derived from the bone marrow monocytes and scattered, that these cells are scattered throughout the stratum spinosum. There may be approximately 800 Langerhans cells per square millimeter. The nuclei and cytoplasm, they have the same structure as in other cell types, but they have specific Birbeck cytoplasmic granules. As you are looking inside of the cytoplasm, they are vermiform granules. They resemble tennis paddles in section, and their main function is phagocytosing activity. These cells appear clear with a light microscope and may be differentiated from the surrounding keratinocytes by the absence of tonofilaments. Similar to the other antigen-presenting cells, Langerhans cells possess Fc and FC fragment receptors for antibodies, and C3 receptors for the complement proteins. They phagocytose antigens and form epitope major compatibility complexes and migrate to nearby lymph nodes where they present the epitope MHC complexes to the T cells. Then the Merkel cells, they derived from the neural crest cells, and they are clear, located in the stratum basale, and especially in oral mucosa, hair follicles, and tips of the fingers. The nuclei of these cells have deep grooves. Their cytoskeleton is rich in cytokeratins, and they are closely linked with the myelinated nerve fibers, as we are looking here. Together, with the nerve fiber, they form Merkel complex. Merkel complex, they function as mechanoreceptors responsible for light touch. Then the melanocytes. The melanocytes are the neural crest derivatives and are located in the stratum basale, as we are looking here. But they have long, slender, finger-like processes that extend into the stratum spinosum, where their tips are surrounded by cytoplasmic extensions of the keratinocytes. Melanocytes possess oval-shaped granules that contain enzyme, the enzyme. Pay attention to this illustration. Here there are processes, and in the cytoplasm we are looking at the granules. Inside of them we have melanin pigment and also tyrosinase enzyme. If we will look at the deeper the mechanism of what is taking place inside of the melanocyte, we will see here that melanosomes migrate into the tips of the melanocyte processes, accumulating more and more melanin along the way. This process is stimulated by the UV radiation. The tips of these melanocyte processes are nipped off by the keratinocytes, and this process is called the cytokine secretion. Pay attention, the tips of these cells will be nipped off by the surrounding keratinocytes. And the melanosomes located within the keratinocytes of the stratum spinosum are attacked by the lysosomal enzymes to be degraded within a few days. The melanin acts to protect the keratinocytes from the UV radiation. Although the population density of melanocytes varies with regions of the body of a single individual, the numbers are essentially the same across the races, and the differences in skin color are due not to a greater number of melanocytes but to the greater production and slower degradation of melanin. Okay, now we will look at the processes which are taking place inside of the cells of the epidermis, inside of the keratinocyte cells. Here we are looking that in the stratum basale, the stratum basale, it is composed of the single layer of cuboidal to low columnar shaped cells. And these cells undergo cell division, and the newly formed cells, they will push the older cells lying above them towards the free surface. And the stratum basale cells form hemidesmosomes with the underlying basement membrane, and the desmosomes with the adjacent cells. The desmosomes and hemidesmosomal plaques have bundles of intermediate filaments. These are tonofilaments. So these tonofilaments begin to be produced within the stratum basale cells. Then the stratum spinosum. Stratum spinosum is a substantial region, and it is composed of several layers of cells that are polyhedral in shape. We will look at the stratum spinosum on the histological section here, the stratum spinosum. And they are polyhedral in shape, these cells, and in the vicinity of the stratum basale, but become flatter as the cells migrate away from the basement membrane. The polyhedral cells display mitotic activity, but cells in the more superficial layer of the stratum spinosum, they no longer divide. And then the organelles of these cells resemble those of the stratum basale. However, the tonofilaments are better developed, especially in the more superficial located flattened cells. They forming thicker bundles known as tonofibrils. In the same region, the flattened cells house secretory granules called membrane-coated granules. Now let's look here. That is the stratum spinosum and tonofilaments. Here on the superficial layer, they will form tonofibrils. Tonofibrils will form, that is the aggregations of tonofilaments. Then lamellar bodies. So membrane coating granules or lamella granules, they contain lamellar deposits of lipids, and inside of them there are glycosphingolipids, phospholipids, ceramide. Then the cytoplasmic extensions of these cells of the stratum spinosum resemble spines, and hence the name of this layer. The spines of adjacent cells interdigitate with each other, and by forming desmosomes, these cells adhere to each other and to cells of the stratum basale and the granulosum. Cells of the stratum granulosum house membrane-coating granules, here, the lamellar granules, and also contain non-membrane bound deposits of keratohyalin. And these keratohyalin granules will contain filaggrin, loricrin, and other proteins. And they will, when they will discharge their content outside, the proteins of their content will stick the tonofilament to each other to form tonofibrils. The contents of membrane coating granules, as we are looking here, and this illustration also, these lamellar granules, they also exocytosed into the extracellular space, superficial to the stratum granulosum and the stratum lucidum, that prevents aqueous fluid from penetrating in either direction. So this one will form a lipid barrier that accumulates between the stratum granulosum and stratum lucidum. The presence of this lipid makes the epidermis impermeable for water. So here, the next layers in stratum lucidum and in the stratum corneum, the horns, the squame cells will stick together via these lipid substances and this green colored region here. So they will stick these cells together and they will form, they will make epidermis impermeable to water, preventing fluid loss from the underlying dermis and the entry of water into the dermis from the outside the body. Then the stratum lucidum, which you cannot see here, is a transparent layer of cells whose organelles, including its nucleus, have been eliminated by the lysosomal enzymes. If you are looking here in the stratum granulosum, there is starting the degenerating of the nucleus. Also the organelles will also degenerate under the influence of lysosomal enzymes. So the internal all the cytoplasmic content of the cell will be disintegrated and the cell become filled with keratin tonofibrils. These cells in stratum lucidum, they are packed with a significant amount of tonofilaments enveloped by ellaidin, its derivative of keratohyalin. The cell membranes of these cells are coated on the cytoplasmic aspect by the protein involucrin, whose function, this involucrin and keratolinin will accumulate under the cell membrane and they will form coat. They will reinforce the cell membrane. So outer surface of the cell membrane will have lipid coat, and the under of the cell membrane also we will have some coat. They will maintain the integrity of the outermost coat of the cell. The stratum corneum, finally, the most superficial layer and is usually the thickest layer of the epidermis of the thick skin. The plasma membrane of these dead cells, known as squames, are thickened and they are filled with keratin filaments. Cells of most superficial layer of the stratum corneum cannot maintain desmosomal contacts with each other, and they cannot maintain any contact with each other. And finally, they will be sloughed off from the surface of the epidermis. And let's look at this slide now. That stratum basale, spinosum, granulosum, lucidum, and the corneum, which is the thickest layer in the epidermis. And here you are looking at the sloughing of these squames from the surface of the epidermis. Now we will talk about the dermis. The dermis, it is underlying component of the skin and it is the dermis, it's connective tissue element of the skin, and it is we can subdivide it into two layers. That is a papillary layer, which abuts the basement membrane and forming evaginations known as dermal ridges that interdigitate with the epidermal ridge, as we are looking here. So here the fibers of loose connective tissue are composed of type 3 collagen and slender elastic fibers that intertwine with one another. Then under the papillary layer we have the reticular layer, which is mainly much denser connective tissue than the papillary layer. It is mainly dense irregular connective tissue, and its fibers are composed mostly of coarse bundles of type 1 collagen interspersed with the thick elastic fibers embedded in a matrix of ground substance rich in dermatan sulfate. Then here inside of the dermis we have a rich plexus of blood vessels and lymph vessels. Then also they will give rise to smaller vessels that supplies a papillary layer, and also is located in the dermis. Encapsulated neural elements such as Pacinian's corpuscles and Ruffini's corpuscles, they respond to deep pressure and tensile forces. And finally, the appendages of the skin. They are the hairs, then the sweat glands. Here we are looking at the sweat gland. Its sector portions located in deep layers of the reticular layer of the dermis, and the sebaceous glands located at the junction between the papillary and reticular layers, and the sweat glands ducts open onto the surface of the epidermis individually, or actually we have two types of sweat glands. They are eccrine and apocrine sweat glands. They produce sweat. And the sebaceous glands ducts open onto the hair follicle, so they produce sebum. And also inside of the skin we have the hairs. We will look at the structure of the hairs, and you know that the hairs, each hair, it is located inside of the hair follicle. It develops from the hair follicle, that is epidermal invaginations that frequently extend into the hypodermis. They are enveloped in a basement membrane known as the glassy membrane. Pay attention, that is a green colored coat which covers this hair follicle, that is a glassy membrane, and that is surrounded by dermally derived connective tissue membrane. So dermal sheet will surround the glassy membrane, so it will be the outermost coat of the hair follicle. Then there are several components of the hair follicle. Hair develops, the hair root, let's look here. Hair root is an enlarged hollow terminus whose concavity is occupied by the vascular connective tissue elements known as a dermal papilla. And these two together are known as hair bulb. The core of the hair root consists of cells known as the matrix. Let's look here at the matrix. Okay, that is the matrix. The mitotic activity of these cells is responsible for hair growth. The core of the hair, okay, then let's look at the immediately deep to the glassy membrane, Here we can see the layer of cells that at the hair bulb that increase the number in the vicinity of the stratum corneum. This layer cells is known as the external root sheet. The internal root sheet surrounded by the external root sheet and is composed of the three layers. Here that is the internal root sheet. So the external or outer root sheet you are looking here by the pink color, and inner root sheet you can see by the purple color. And here we can distinguish three layers. What are those layers? First of all, the Henle's layer, outermost, then Huxley's layer here, that is middle layer, and finally the cuticle, that is this one. So these three components, they compose the internal root sheet. Then The internal root sheet develops from the most peripheral cells of the matrix. Pay attention to this region. And it is if the internal root sheet develops from the outermost layers of the dermal matrix, so that hair matrix. So the external root sheet or outer root sheet, it develops from the stratum basale of the epidermis of the skin. Then the internal root sheet extends from the matrix to the region where the duct of the sebaceous gland enters the hair canal. The absence of the internal root sheet from that point leaves a space known as the canal of hair follicle. The hair shaft, here you are looking, hair shaft is a part of the hair follicle that extends through the epidermis, and it has 3 layers. What are those layers? Most peripheral is the cuticle of the hair. Don't mix it with the cuticle of the internal root sheet. So cuticle of the hair, you are looking here. Then the cortex and the medulla here. They will form the hair shaft. And so that okay, we have to understand this during the migration away from the hair matrix, the cells that arise from the peripheral cells of the hair matrix, when they move, they migrate from the site of origin. These cells of the cortex manufacture and accumulate keratin filaments that become embedded in a matrix of trichohyalin, a substance similar to the keratohyalin of the stratum granulosum in the epidermis, and form the hard keratin that is characteristic of the hair shaft. So it is unlike the soft keratin that is present in the epidermis. The hairs include hard keratin, so as the nails too. The central core of the hair shaft, that is the medulla, arises from the most central cells of the matrix. The medulla is displaced by the cells of the cortex as the hair shaft extends above the skin surface. And now let's talk about something about the sebaceous gland structure. Here you will see the histological section of the sebaceous glands. Sebaceous glands, also called holocrine glands, and they are associated with the hair follicles. And as I told you, here you can see the hair, and the ducts of the sebaceous gland open into the hair follicle. The oily sebum that is product of this gland, enters the hair follicle. And the most peripheral cells, they are actually simple branched. These glands are simple branched alveolar glands. Here you can see their secretor portions. And the most peripheral cells of these globular glands, most peripheral cells, they are flattened and they undergo mitotic division. The larger cells are centrally located and they accumulate lipid droplets that eventually displace the organelles of the cells, causing their degeneration, necrosis, and transformation into sebum that coats the hair shaft and the skin surface after releasing outside. Sebum makes the hair less brittle and the skin more supple. So sebaceous glands, similar to apocrine sweat glands, are under hormonal control and become more active after puberty and may lead to formation of the various acne. Okay, that is about the skin and skin appendage. Now let's go to the respiratory system. The respiratory system, the lungs and the airways leading to and from the lungs, distributes oxygen and it removes the carbon dioxide from the cells of the body. The ability of the respiratory system to accomplish these essential responsibilities depends on, first of all, ventilation, the breathing that propels air to and from the lungs. Then external respiration, it's transferring in the bloodstream the inhaled oxygen for the carbon dioxide released by cells. Then oxygen and carbon dioxide delivery, and then internal respiration. It's exchange of oxygen for carbon dioxide in the cellular environment. So ventilation and external respiration are the domains of the respiratory system. The delivery of oxygen and carbon dioxide is a function of the circulatory system, and internal respiration is a cellular event that occurs in the vicinity of all living cells. The proper function of the respiratory system requires that the inspired air be delivered by the conducting portion to the respiratory portion where exchange of gases can occur. The conducting portion of the respiratory system consists of the nasal cavity, mouth, nasopharynx, pharynx, larynx, trachea, primary bronchi, then secondary bronchi, finally bronchioles, and terminal bronchioles. And so in the terminal bronchioles, the conducting system of the respiratory system is finishes. And as the passageways branch and get closer to the respiratory portion, they decrease in diameter but increase in number. The total cross-sectional area increase at deeper levels, causing a decrease in the velocity of airflow as the inspired air approaches its final destination, the alveolus. So the nasal cavity begins at the nostrils, or let's look at this illustration. Okay, at the nostrils, and that ends at the choana. Okay, as you are looking here, and it is divided into two halves by the bony and cartilaginous nasal septum. That's anterior aspect, which is lined by the skin with vibrissae, which filter large particulate matter present in the inspired air. And the pseudostratified columnar epithelium of the posterior aspect of the nasal cavity is rich in goblet cells. The underlying connective tissue has an abundant vascular supply with large venous sinusoids, ceromucous glands, and rich supply of lymphoid cells and antibodies. It also has olfactory region, olfactory mucosa, as you can see here, and it's located in the posterior superior aspect of the nasal cavity. It is yellowish and houses the olfactory epithelium that perceives odors. Cells of the olfactory epithelium there will include basal, sustentacular, and olfactory cells that you studied in the sensory system topic. Now we'll look at the next illustration. Here we can see the branching pattern of the airways. As we are looking here, the trachea it bifurcates and it signals the beginning of the bronchial tree here. That conducting portion of the respiratory system, which extends all the way into the lungs down the level to the level of the terminal bronchioles. And as the branches of the bronchial tree bifurcate, their diameter decreases and the number increases. And as I just told you, the total cross-sectional area of the bronchial tree increase in size, slowing the velocity of the inspired airflow and increasing the velocity of expired airflow. The bronchial tree comprises, pay attention, the primary bronchi. Here you can see, and then they subdivide into second primary bronchi. They are main bronchi also called. Then here we are looking at the main bronchus. It's written here. It subdivides into secondary bronchi. Secondary bronchi or lobar bronchi. As we know, in right lung we have 3 lobes. That's why in the right side we will have 3 lobar or secondary bronchi. In left side we will have just 2 of them. Then they undergo subdivision into segmentar or tertiary bronchi. So the lobar bronchi will subdivide into tertiary bronchi. So the tertiary bronchi continue the branching pattern till they lead to the bronchioles. Pay attention to the bronchiole, they are smaller bronchi. And finally, they come to the terminal bronchiole. Here you can see it. So at the terminal bronchiole, the respiratory portion, the conducting portion of the respiratory system is finishes. And here starts the respiratory portion. It starts with the respiratory bronchioles, then it will branch into alveolar ducts. They will branch till the alveolar sacs, and in the wall we will see alveoli, the sac-like structures with thin wall, which will provide the gas exchange. And so here I will tell you something concerning the histology of the bronchial tree as it progresses from the primary bronchi and till the bronchioles, till the terminal bronchioles. Here, as you can see here, the size of cartilage and glands will decrease, and then the percent of goblet cells and the epithelial thickness decrease. Then the amount of elastic tissue and smooth muscle relative to the thickness of its wall will increase, as here we can see elastic fibers and smooth muscles we will find in the wall of the respiratory portion also. So if we are talking about each of these components, we'll look at this one. And here, so the trachea, we'll look first of all at the structure of the trachea. Here you can see the trachea. It is the tube that is always patent because of the 10 to 12 C-shaped hyaline cartilage rings called C rings that support it along its entire length. Each C ring positioned in such a fashion that its closed portion faces in an anterior direction has its own perichondrium. The perichondria of neighboring C rings are connected to each other by fibroelastic connective tissue. The posteriorly facing open ends of the C rings are connected to each other by slips of smooth muscles, that is trachealis muscle, here you can see, whose contraction reduces the diameter of the trachea, accelerating the flow of air through this tubular structure. The lumen of the trachea resembles the shape of the capital letter D, as we are looking here. The trachea is composed of 3 layers: the innermost mucosa, pay attention, which is trachea that is composed of the respiratory epithelium and the subepithelial connective tissue, and a thick sheet of elastic fibers that separates the mucosa from the submucosa. The respiratory epithelium pseudostratified ciliated columnar epithelium, and it is composed of the several types of cells. Under the submucosa, we are looking at the cartilage. Here you are looking at the cartilage part. Then the outermost coat will be the adventitia. Adventitia will adhere the trachea to the surrounding tissues. Now let's look at the cells which are present in the respiratory epithelium. Here almost 1/3 of cells of this epithelium are goblet cells. Pay attention, these are goblet cells here. These are unicellular glands that manufacture the slippery secretion known as mucinogen, which they store in secretory vesicles. And then there are approximately as many ciliated columnar cells. Here you are looking at the ciliated cells, and these tall cells, they have the cilia on their surface. Pay attention, on their apical surface we have a lot of cilia that propel the mucus in the lumen of the trachea towards the larynx. The basal cells, pay attention, they are short regenerative cells. They constitute slightly less than 1/3 of the cell population of the respiratory epithelium. These cells undergo mitosis and replace the dead and dying goblet cells, ciliated cells, and brush cells. Then the brush cells, and they are known as small granule cells. They constitute less than 3% of the total cell population of the respiratory epithelium. These narrow cells may be goblet cells that have released the mucinogenes, or they may have some neurosecretory function because they have long microvilli that extend into the lumen of the trachea. Then we are coming to the serous cells. These are approximately also 3% they constitute. Serous cells are also tall columnar cells that display apical secretory granules containing a serous secretion whose function also not exactly understood by now. Then the trachea include DNAS cells. These are diffuse neuroendocrine cells, and these cells also compose less than 3% of the total cell population. These cells manufacture and release paracrine and endocrine hormones in response to stimuli such as hypoxia. DNAS cells are frequently innervated, and the nerve fiber DNA cells complex is known as pulmonary neuroepithelial bodies, which under hypoxic conditions may be able to activate neurons in the respiratory center of the hypothalamus. Then underlying connective tissue will form lamina propria of the trachea, and then we will have the next list that will be submucosa and the adventitia. Now let's come to the structure of the bronchus. Actually, we know the bronchial tree include different bronchi. The primary, secondary, tertiary, but their general principle of structural organization is almost the same. For example, the secondary bronchi. In the secondary bronchi, they resemble the primary bronchi, but the supporting hyaline cartilage is no longer in the shape of a C-ring. Instead, it is composed of small cartilage pieces that surround the circumference of the lumen. Here, for example, that is short of the cartilage, but in secondary bronchi, they will have not completely C-shape, but partially it will surround the wall. And because the cartilage no longer has open arms facing in a posterior direction, the smooth muscle migrates towards the lumen, occupying a position between the lamina propria and the submucosa, and it is arranged into helically disposed layers. The fibroelastic adventitia has elastic fibers radiating from it in such a fashion that the fibers are positioned more and or less perpendicularly, and they will contact with the wall of the secondary bronchi. Then the secondary bronchi, when in the lung, aborrise to form tertiary, and each of which serves 1 to 10 relatively large bronchopulmonary segments of each lung. And if we are coming, so here we will see also the components of mucosa, the respiratory epithelium, which include the same content and as in the trachea. Then underlying lamina propria, then submucosa we have, and as just I mentioned, that is adventitia. And then here we are looking at the respiratory epithelium in scanning electron micrograph representation. Here we can see the cilia, then the goblet cells. Here on the surface we can find a little amount of the microvilli. Then the epithelium, lamina propria here, then the submucosa. And here we also have to mention that lamina propria of the respiratory mucosa, it includes glands. They are seromucous glands. They release content onto the surface of the epithelium, and it will form the mucus on the surface of it. And by propelling of the cilia, the mucus will move towards the oropharynx. By that way, the bronchial tree will clean itself. So, and we will come to the bronchioles here. And what are the bronchioles? These are the small bronchi, and in the wall they will have no cartilage, then they will have relatively thick smooth muscle coat compared with the thickness of their wall. The elastic fibers that emanate from the connective tissue enveloping the smooth muscle bundles in such fashion that they are positioned more or less perpendicular to a tangent placed at the contact with the wall of the bronchiole. Contact with other elastic fibers arising from the various sources, placing tension on the entire circumference of the bronchiole and maintaining its patency. Then the bronchiole has no glands in the lamina propria. Then a simple columnar ciliated to a simple cuboidal ciliated epithelial lining it will have. So instead of stratified ciliated epithelium, here we will find the simple columnar to simple cuboidal epithelium. Then the epithelium lining of this bronchiole will not have goblet cells, but in smaller bronchioles will arise Clara cells, and they will secrete surfactant-like substance that assists in maintaining bronchiolar patency, and they will destroy the inhaled toxins and act as regenerative. They will act, pay attention, here we are looking at them, at these cells which covers the bronchioles, the epithelium simple cuboidal. Here you are looking the ciliated cells, pay attention, and also the brush cells which have a little amount of the microvilli, and the Clara cells which produce these surfactant-like substances, and they also destroy inhaled toxins, and also they act as regenerative cells to maintain the epithelial lining. Bronchioles arise to give rise to terminal bronchioles, the smallest segments of the conducting portion of the respiratory system, each of which serve as small segment to a long lobule, the lung lobule, that is a lung acinus. The terminal bronchioles resemble bronchioles but are much more slender and are lined by simple epithelium composed of the just cuboidal cells and the Clara cells. Their narrow lamina propria is surrounded by some smooth muscle cells, and let's look here the continuation of the terminal bronchioles, and they will lead to the respiratory portion of the respiratory system. The respiratory portion of the respiratory system will start with the respiratory bronchioles, and the respiratory bronchioles resemble terminal bronchioles with the exception of the occasional alveoli that balloon out of the wall. And so in the wall of the respiratory bronchioles, we will have also alveoli, and their walls permit the exchange of gases to occur, which does not happen in terminal bronchioles. Respiratory bronchioles undergo branching and eventually give rise to alveolar ducts. Pay attention to the alveolar ducts here. That is a respiratory bronchiole. In the wall, it has alveolus, as we are looking here. Then it leads to alveolar ducts. One of the alveolar duct here, other one here. Then the alveolar ducts, they simply line a sequence of alveoli that branch to form many additional alveolar ducts. Each of these alveolar ducts ends as a they will continue into the alveolar sacs. Pay attention. They will branch into the alveolar sacs. Here we are looking. The common air space from which these groups of alveolar sacs originate is known as atrium. Alveolar ducts, alveolar sacs, and alveoli possess their own basal lamina, and they maintain their patency by the presence of slender elastic fibers that attach to the slight connective tissue elements of these structures and to other elastic fibers in the vicinity. So what are the alveoli? Let's look here. The alveoli are small air spaces lined by two types of cells, which are, we will look on next illustrations at these structures. For example, terminal bronchioles here, respiratory bronchiole, and alveolar duct, which lead to alveolar sacs And here in the walls we can see the many alveoli. And in the illustration also, let's look. There are terminal bronchiole, respiratory bronchiole, then the alveolar duct and alveolar sacs. They are alveoli, so thin walled sac-like structures. Now we are looking at the wall of the alveoli. So as I mentioned, there are two types of cells. First of all, type 1 pneumocyte or alveolocytes. Here they are highly attenuated, squamous in shape. Then we have type 2 pneumocytes. Type 2 pneumocyte, these are or alveolocytes, which have yellow color here. And they are also called septal cells. The very thin wall of the alveolus permits the exchange of oxygen for carbon dioxide. A smooth muscle cell and its surrounding reticular fibers encircle the opening of each alveolus, controlling the size of its aperture. It has been estimated that there are 300 million alveoli in the two lungs, where each alveolus is approximately 0.2 subdivided into the 1,000 quadrat millimeters, providing a total surface area of 140 quadrat meters devoted to the exchange of oxygen for carbon dioxide. Type 1 pneumocyte, as we are looking here, they form greater than 95% of alveolar surface. Most of the organelles of this cell are crowded around the region of the nucleus, whereas the remaining region of the cell are approximately 8 nanometers in thickness and house mostly the fluid cytosol. Adjacent type 1 pneumocytes form occluding junctions with each other to avoid the entry of extracellular fluid into the surrounding tissues. Then also this they will provide exchange of gases. But type 2 pneumocytes, they are cuboidal shaped cells. They form just 5% of the alveolar surface, even though they outnumber the type 1 pneumocytes. And these cells house lamellar bodies in the cytoplasm. They discharge them into the lumen of the alveolus as pulmonary surfactant, and it is a substance composed of surfactant proteins and phosphatidylcholine substances. They will coat the wall of the alveolar airspace, and by reducing surface tension, it assists in maintaining alveolar patency. These cells not only manufacture and resorb surfactant in continuous manner, but also they have the ability to enter the cell cycle and form more type 2 and type 1 pneumocytes. So these are also regenerative cells. And then the region between the neighboring alveoli is known as interalveolar septum, which is slender, continuous, capillary-rich connective tissue. Here you are looking at the capillaries. So here that is a connective tissue that provides a degree of stability to these delicate structures. Here we can find monocytes, then alveolar macrophages, which called dust cells, and here you can see these macrophages, which phagocytose not only particular matter and microorganisms that gain access to the alveoli with the inhaled air, but also surfactant to ensure a constant exchange of old for newly formed surfactant. Dust cells injected with phagocytosed material either enter the interalveolar septum, to leave the lung via lymph vessels, or migrate up the bronchial tree. They migrate up the bronchial tree to enter the pharynx to be eliminated with the mucus by being swallowed or expectorated. So now we are coming to the let's look at these illustrations. Here the electron-micrograph representation of the alveolar wall of the alveoli. Here in the wall we can see the blood capillary And here we can see the portion of the alveolus which is in close contact with the capillary wall. And here by high magnification of the light microscope, you can see the wall of the alveolus, and its wall contain blood capillaries. Here we can also distinguish them, and and the regions where they will come, these walls of the capillaries and wall of the alveolus, they come in contact with each other. There they will form the blood-air barrier or blood-gas barrier. It is composed of the capillary endothelium, that is the capillary lumen with red blood cell in its lumen. And here you can see that is the alveolus, the lumen of the alveolus. So here we will see exchange, here will take place gas exchange. The blood-air barrier, it contains some main components. They are, first of all, capillary endothelium. Pay attention. Then fused basal lamina of the capillary endothelium and type 1st pneumocyte. Here that is the type 1st pneumocyte. Here that covers the alveolus wall. Then attenuated type 1st pneumocyte cytoplasm and the surfactant which covers the surface of the first type of pneumocyte. So these structures will form the blood-gas barrier, blood-air barrier, which provide exchange of gases needed for the exchange of the gases in the body. And here we can see that, pay attention, the section of the lungs with the components in the lungs, that is, we have to know the connective tissue elements here and the alveolus wall, the lumen of the alveoli. The interalveolar septum may be very narrow actually, and housing only continuous capillary whose basal lamina is fused with a basal lamina of the type 1st pneumocyte, or a capillary and connective tissue elements including lymphoid cells, fibroblasts, and macrophages that surrounded by the reticular fibers, elastic fibers, ground substance, and extracellular fluid. So that's all for today's lecture. Thank you for your attention.